Thank you, Eugenio. It gives me great pleasure to introduce our first speaker, Ada Jonat. She's going to talk about light at the end of the ribosome tunnel. He focuses on protein biosynthesis and antibiotics paralyzing it. She graduated from the Hebrew University, earned her PhD from the Weizmann Institute, and completed postdoc work at Mellon Institute and MIT in the US. In the 70s, she established the first structural biology laboratory in Israel. She's currently the director of the Kimmelman Center for Biomolecular Structure at the Weizmann Institute. Previously, she also headed the Max Planck Research Unit for Ribosome Structure in Hamburg. Among many others, she is a member of the US National Academy of Sciences, the Israeli Academy of Sciences and Humanities, and was appointed to the Pontifical Academy of Science by Pope Francis last October. She holds honorary degrees, doctorates from over 20 universities, and her awards include the Israel Prize, the Linus Pauling Gold Medal, the Wolf Prize, the Albert Einstein World Award for Excellence, the Arichi Peace Prize, and of course the 2009 Nobel Prize for Chemistry. Ada Jonat.
Questa anche.
I apologize for standing here. I'm an old-fashioned, you can see, and I'm using some softwares that are out of date, but are good for me. So first, thank you for inviting me. I don't exactly know why I fit into here. But when I found out, I didn't know, when I found out that this conference talks about light as coming from lasers, but also as coming from upstairs or above us, or as they inspire intellectuals, I decided to use the known phrase, light at the end of the tunnel, that was used in wars and so on, but here it's the ribosome tunnel. I hope that all of you know what are proteins. Just for those that don't remember, they are doing almost everything that cells need. And cells, living cells, is not only human cells, it's also bacteria and elephants and cockroaches and plants. What I have here are some examples for human ribosomes, like receptors. Enzymes that are chewing and cutting what we eat. Hemoglobin that transports oxygen, regulators like insulin and skin and hair, they are all proteins, structural proteins in this case. So in brief, in conclusion or in summary, the proteins are responsible for all what happens to us in smooth life and also under pressure, like immune response. Sometimes in several heart events, the two enzymes can take care of it. Dust in the eye, the tears contain a protein that can chew the walls of every bacteria so they can eliminate or limit eye infections. And so proteins are long chains made of amino acids. There are 20 of them. And the fold of each protein allows its function and is dictated by the sequence of the protein, of the amino acids that make it. There are about, in normal proteins, about 150 to 500 Amino acids, there are some smaller and some larger, but that's average. Just to tell you what I mean by the fold structure and function, you can have a look at my example, which is talking about a very complicated and innovating machinery, paper clips, that can keep together several pieces of paper. With just unfolding them, not putting them in acid or base or cut or heat or whatever, just unfolding them like most of the nervous people are doing when they talk. We get the same material, but with no fold that allows its function. And this is correct also for proteins, and I want to show those of you that never saw a representation of a protein according to its structure. Proteins are made of, typically, of nitrogen, hydrogen, carbon, and sulfur, a little bit sulfur. They have a very complicated structure that has several tasks, but the main task is to keep the active side of it, where things are happening. You see here, there is a groove where the things are happening, and we call it active side. I rotated it and enlarged it a bit, so you can see it in more detail, with its substrate that fits exactly into the active side. So the substrate can be cut. For instance, in this case, this is a cutting protein, an enzyme. And that's dictated by the sequence. And the sequence is dictated by the gene that is coding for the specific protein. Each protein has a gene. Each gene has a protein. There are proteins that are common to the same organism, like the proteins I showed earlier. And of course, there are specific proteins that are according to the person, like bald hair, white hair, black hair, and so on. So this actually, if you allow me to tell you what children told me, maybe you enjoy it. When I ask children, I'm talking a lot to school kids, 6, 7 years old. When I ask them, do you know what is DNA? Yeah, sure, we know. And so what is it? Because when I was 6 or 7, it was just discovered. What is it? This is the tool that the police is using to identify criminals. So these are the specific genes. But anyway, each protein has a gene. The genes are in the DNA. The DNA is all the genes together. It's a language of 4 letters, and each 3 of them are coding for a specific amino acid. So each amino acid has at least one triplet that codes for it. So the translation of the genetic code means how we go from the language of instructions to the product. And this excited me and drew my curiosity since I was a school kid. And as I told you, DNA was only found out, and when I became a student, the triplets were already assigned. So I wanted to understand how things are happening. So now we know DNA is a double helical structure. The letters are here, in each of them is called base in base pairs, and they are not available. They are covered by the structure, by the double helical structure. So first of all, they are being transcribed to a very similar molecule called messenger RNA, or RNA in general. RNA is like DNA, the backbone is different. The 4 bases are, 3 of them are exactly the same, 1 of them is slightly different, but from the chemical point of view, it doesn't matter. And this is available, can be translated to the growing protein by a cellular machinery, the green one called the ribosome. And what I wanted to understand is how this happens. There was a lot of dispute about the possibility to do it. I was not sure when I started, but now we know. So now we can describe the ribosome as a factory, and this factory specifically makes these long chains. These are proteins. The instructions are coming in on messenger RNA, the genetic instructions. They are being decoded here. And at the same time, each carrier that has this triplet also carries has this anti-triplet that can make base pairs with the triplet. carries the cognate amino acid to it. So then when this triplet will come in, this amino acid will be bound inside to the growing protein, and then the drug can go out. Please forgive me, I don't know to grow inside, so I grow outside, but it should be inside. And this costs energy, at least two energy-rich molecules, in this case GTP, are used for each of the elongation cycles. So, how did I get into it? I can't tell you the whole story because it's much too long. I have a time limit. But I can tell you that I read, after having an accident, bicycle accident, I had a lot of time and I read about the bears that go to sleep, to hibernate at the beginning of the winter, and they get up. And for instant recovery, for the things they have to do when they get up, they need proteins. And for having proteins, they need ribosomes. Ribosomes in general are not living long time. They are deteriorating every 3 or 4 days in the body, in the fridge, in the test tube. Has to be made new. The cells are making them all the time, but when the bear is sleeping, there is much less activity. Many ribosomes could have been deteriorated if the cell wouldn't do something to preserve them. And it was found that ribosomes are well packed, orderly, in monolayers, like a two-dimensional crystals, just one layer inside the cells. And I thought that this is the way that nature, the mechanism that nature provides the bears to get up and immediately start functioning, because these ribosomes can make the proteins. So, just a minute. So not only me read this story, some other groups, and they decided that freezing or cold is inducing order, because earlier many very good, fantastic groups, very leading scientists tried to crystallize ribosomes and failed. But bears are doing it, at least in monolayers. So they decided that this cold stress is inducing packing, and they used fertilized eggs of chicken or of lizard, put them in the fridge, and after half an hour they had the ribosomes organized, and this is the organization that was found. Actually very nice, but limited, only of membranes and only of eukaryotes. I thought that every stress should induce periodicity, because under stress, ribosomes or the cells are feeling maybe the stress will go away and life will come back. So I decided to use ribosomes from bacteria. I actually wanted bacteria, and you will see later why bacteria and not eukaryotes, that grow under stress, like in the Dead Sea. For all of you that were not at the Dead Sea, I hope that many of you did. It's in Israel, it's the lowest point in the world. It's called dead because there is no life. Not even one flower or one fish or whatever. And you can see it from a close view. These are salt crystals that come out every year while it is being evaporated, from the winter until the summer, so many. You can see that although it's called Dead Sea, there is life in this Dead Sea, very little life, but there is. Here is a colony of bacteria called Haloarcula maris morti. I think that in Italy I don't have to explain what is maris morti. This is one type, and they grow even on the salt. So there was some evaporation, and you can see them here. You can see even better in here. And what you see here, these pinkies, this is several colonies, and the level from here to here is 78 centimeters that was evaporated from January to September when this picture was taken. We also used another bacteria called the Anococcus radiodurans. Here it is. This is one cell of it that can live under every super extreme conditions, not only high salt, not only heat. Those are also exposed to very high temperatures, 50 to 60 degrees. But these have 4 rooms. One room is always functioning, the others are storage, and it can live even under very high radiation. So instead of talking much, we got crystals, which are very nice. And I want to show you what we know today about ribosomes. What you will see here is a clip, a movie that I made together with 2 students of the Art Academy in Jerusalem from results that were obtained in several labs, all crystallographic. The movie that should come in a minute is available on YouTube in case here it comes. And it shows, as I say, the iteration of several structures, each of them capturing the ribosome under different functional state. And as it is in YouTube, and many, over 100,000 of people already looked at it, and nobody explains. I hope that what we show is the consensus structure. What you see here are ribosomes from bacteria. So here, messenger RNA is reaching the small ribosomal subunit. Each ribosome has 2 subunits and attaches itself to it. Here, the decoding happens. So the small subunit is the one that organizes the decoding and also makes sure it has a very high proof reading machinery, and also makes sure that it's okay. What you see here, the blue thing is initiation factor. Bacteria have 3 of them. They tell ribosome start when the cell needs them. So here it comes. It sits, everything is okay. So the first tRNA can be brought by another initiation factor 2, is bluish, and bind itself. And now the 2 initiation factors make sure that the binding is correct. You see the tRNA and the amino acid bound to it. So the large subunit can come, associate base of surface complementarity, and also make bridges by conformational changes. Now you can see the ribosome in action. The whole ribosome from the large subunit side. Elongation factors are bringing in tRNAs. The tRNAs are coming with amino acid, going out empty, and the bond is being made in the large subunit at the same time, real quick, 40 in a second. And I want to show you just that the newly bound protein is getting out of the ribosome through a tunnel. That's the tunnel in the title. You see the tunnel? So the newly bound protein, the bond is being made here, will come out through a tunnel. You see now the whole ribosome, the end of the tunnel is at the end of the other side of the large subunit. The process continues and continues and continues until the gene has a stop codon. Once there is a stop codon, instead of new amino acid come factors like this yellow one, which are release factors or recycling factors, and they let the 2 subunits separate and the protein coming out. Okay, so that's the end of the tunnel for the protein. So just for you, for all of you that are interested in more detail, we now know the position of each and every atom in the small and large subunit in bacteria and in eukaryotes. I'm showing here the first one, the bacteria. It's made of many components, RNA and many proteins. The RNA is in gray, and the large subunit, the same color code. So we can tell you about each and every atom, and I want to focus on the tunnel. So in the 60s, in 1967, Malkin and Rich, Alex Rich died 3 weeks ago, sorry. They saw that there is a partial resistance of the newly bound protein when it comes out of the ribosome. Not all the protein is being chewed by proteolytic enzymes. And in 1970, it was shown even in more detail for specific proteins by Global and Sabatini that ribosome protects the newly bound protein. You already know about the tunnel, but at that time, nobody knew, and these 2 papers were filed in the garbage or below everything. There was no except for these two, I didn't find any mentioning of them in any review. But in 20 years later, in '86, together with Kevin Leonard from the EMBL, we saw a tunnel by looking at sections that are, two-dimensional sheets that we learned to get from bacteria. So these are the sheets, and you can see here, these are filter sheets, and you can see here entrance to the tunnel. Actually, it's not the best picture, and I will show you a better one. But first of all, I want to tell you, everybody was against it, because nobody knew about it, and they thought that the ribosomes coming on the surface of the ribosome. So even Peter Moore said, we have to check this, it's only maybe, okay, occasional or whatever. 9 years later, the tunnel, well, there was a story about tunnel or channel, I can tell you later, not formally, came out. And now there are many papers talking about it. I don't want to tell about them, just to show you. All these are discussing what happens to the tunnel. Even Johnson even thinks that the tunnel opens when there are membrane proteins, hydrophobic proteins being made. So there are 103, there were 103 papers about it 5 years ago. More. Folding zones, more and more and more. I really don't want to talk about all of them. I just want to show you that the tunnel exists, and here is part of it, as we found in our crystallographic studies. Here the tunnel, here is a newly born protein. Here it can be folded, and here is where antibiotics for the most known family bind. So not only there is a tunnel, not only the proteins go through it, it is also providing a pocket for macrolides, which are the children and grandchildren and cousins of erythromycin, the first ribosomal antibiotic that is being used until now in hospitals. So this is the folding zone, and I don't want to talk about this. I want to talk more about the antibiotics. So over 40% of the useful antibiotics bind to ribosomes, paralyze ribosomes. And as I said, most of them are binding to the tunnel. So what are the natural antibiotics? They are the weapons that the microorganisms are using each against other. It means they invented wars, not us, before us. And the question that we ask, not only us, how do the ribosomes, which are usually less than 1,000 Dalton in weight, can paralyze ribosomes? Ribosomes from bacteria are of 2.5 million Dalton molecular weight. So the trick is like David against Goliath, going to exposed functional places. So what you see here are the two skeletons of the large and the small subunit, and the balls are where families of antibiotics bind. So all of them, or most of them, bind to functional sites. This to the decoding in the small subunit, this to bonding in the large subunit, and the tunnel in the large subunit, and here to the hinging. You remember in the movie this motion? If they bind here, they stop the motion, so that's it. That's it? You don't want to see antibiotics? So I think that even if I have to finish now, I must show you 1.5 minute movie. Oh, 5 minutes, fantastic. So this is an antibiotic that stops the progression of the messenger. It's called Edein. It's fully universal. It means the patients, human patients, vet patients like cows and so on, are killed by it, not only bacteria, so it's not allowed to use it, but its mechanism is really interesting. You see, you don't have to be large in order to stop. The second one is tetracycline that binds to the second position of the tRNA, just occupies the positions, that's all. So the tRNA that brings the amino acid cannot bind. The third one is erythromycin that is binding to the tunnel, and it's the first one that went into clinical use, and I mentioned it already. So here it goes into the tunnel, and only very, very short protein can be made, 5 to 7 bonds. That's it. Blocks. And the last one is the most clever chemically, which is called clindamycin, and goes into the bond. Look here, the bond. You remember? Check. That's it. No bond. Very clever. So I wanted to talk about many more things, but I was told yesterday, at the end of the day, and then I looked at the program very carefully, and I saw that there is a lecture about light on the beginning of life. So I want to tell you about it, what we found. Here. So what we found, hello, be clever, huh? What we found is both functions of ribosomes, the decoding and peptide bond formation, are produced by RNA. Proteins are making everything, almost everything the cell needs, but not themselves. So if I go back to children and I tell them, what do you mean by, what do you think about this? Why proteins are doing everything but not themselves? The children say, so there will not be bias. If protein can make itself, it will make more from himself or his friends. And it sounds funny, but no. I think that this is the way that the cells, nature produces for regulation. One kingdom is proteins, and another kingdom makes them, RNA in the ribosome. So this is in good agreement with the composition of ribosomes. 2/3 of them are RNA, and also with the idea that there was a prebiotic world dominated by RNA. So we started to look for some evidence for it, and we found that around the position where peptide bond is being made, there is full conservation. Full conservation. This piece, where protein is being made, is fully conserved in all known structures and fully conserved from the point of view of segments in every ribosome from every kingdom, 930 kingdoms, ribosomes we checked. If we put the fold of them, one on top of the other, just the fold, not the sequence, they are exactly the same. And you see here, eubacteria, archaea, eukaryotes, mitochondria, they're all the same, exactly. You cannot see the difference. Peptide bond will be made here. And if you know, these are the references from where we took the coordinates. So this high conservation, and it's semi-symmetrical, as you can see, it accommodates the two tRNAs. This high conservation of the semi-symmetrical region indicates its existence beyond environmental conditions. So we called it the proto-ribosome. This is our proposed proto-ribosome, which means that the prebiotic RNA apparatus, this catalytic capability, is functioning between us million times a day. Or in other words, it agrees with the idea of fossil, molecular fossils. So I just want to show you the size of it. You see rotating the large subunit, here it is. And this is the way it looks, I'm finishing. And this is the way from the side that I showed you earlier. So, oops. So based on the suggested existence of a world before us, dominated by RNA, and all the finding that RNA can replicate and elongate itself and can have catalytic capabilities, we propose that the proto-ribosome is the entity around which life evolved. We're trying to show it. One of our hypotheses that it was made by dimerization, so we make the pieces of RNA like in the ribosome today or like others. Oops, sorry. And we found, doesn't matter what we are using as RNA chains of the right size, right length, there is a non-uniform tendency to dimerize. Only half of them more or less dimerized, and this shows that we are looking for pre-Darwinism. So let's forget it. I just tell you the summary. We found that within the complex ribosome structure, very complex, and now we know also larger ribosomes, a region which is 3 to 4% of the total ribosomal RNA shows to us a origin of life. So these studies on ribosomes started with strong collaboration with Professor Wittmann from Berlin, Max Planck. He died 10 years before the structures and was replaced by Franceschi and then by Fucini. She's Italian. The Weizmann Institute is where I'm working, as you heard before. The president decided that I can stay there, although on my entity was called DREAM, was Michael Sena, who was replaced by Chaim Harari, a physicist. They did not change it. It was supported by the committee, by the advisory committee. I had 2 research groups, one in Hamburg and one in Israel. You heard about them too, and I want to thank them. This is a group in Hamburg that terminated 11 years ago, but you can see the TAs, the technical technicians, call themselves Angel, and the group members came to the Dead Sea to look for better bacteria. They had better life. This is a group in Israel that is still existing. It is being run actually not by me, but the senior scientist is Dr. Anat Bashan, and the person that found the symmetry in the ribosome is and suggested the proto-ribosome is Ilana Amon. Tamar came to us 16 years ago for 10 weeks. She's still there. She is a really good housewife and now a mother of 3, and she made a cake for her birthday. And this is her cake. So it shows that in my group, ribosomes are considered sweet. I want to show you my family that was very supportive all the time. My mother, I'm not showing here, my sister and so on, but my daughter, Hagit, she's an MD, and her daughter, who is now here a school kid, and she read about me when I got the prize in Paris. As you know, she is a very busy scientist, but she always finds time for me. This is what she thought, and she gave me a prize. This is the Nobel medal, and this is why you invited me here. But for me, second important, and maybe more, is this prize. The Grandma of the Year is Ada Yonat, that my granddaughter gave me before the Nobel. So when I asked her which year, she said, which year? Every year, you have to reprove yourself. And if you fail, I'll take it off the wall, and it's still on the wall. So ribosomes became popular in Israel. You see, here's me, and she is dancing with her. She's a ribosome. Protein comes out of her. And even young children became ribosome, and this is what happened to me. And if you look carefully, small subunit and large subunit, and the artist that made it, Michel Kishka, never met me before, and he's not a crystallographer. And thank you very much.
We have a short time for questions.
It's Vince Cerf again from Google. I'm willing to ask a short question. I have no idea if there's a short answer. Do the ribosomes, they are created like all the other things that are inside the cell from the generation of proteins through interpretation of DNA. Is it the case that all the pieces that are needed to create the ribosome are found inside the cell nucleus? Or is this a more complicated process to create additional ribosomes inside the cell?
I'm trying to answer this now.
You need to get the microphone again, sorry.
So first of all, not all cells have nucleus. We talk about bacteria, they don't even have it. But all proteins that are needed for creating ribosomes are made by ribosomes. The ribosome RNA is made by the RNA machinery. This is correct for every cell, and it doesn't matter where it sits. In mitochondria, it's even more complicated, but doesn't matter. The cell has a mechanism to make the ribosomes all the time. They are living 3 or 4 days in us all the time, and they are working all the time. I didn't talk, but they make a peptide bond. They can make 40 peptide bonds in 1 second. I needed 6 hours for one. I'm being a good student.
Thank you.
Okay, hearing no more questions, I think we will thank Dr. Junat again. Our next speaker is Dr. Albert Brugge, and his title is Per Aspera ad Astra, towards artificial photosynthesis. The Latin part of his title actually happens to be the motto of the state of Kansas in the United States. He's a master in organic chemistry at the University of La Sapienza and a PhD in University of Twente in the Netherlands. After a time as a postdoc in the group of Professor K. Severin at EPFL, he is now a lecturer at the University of Freiburg and is doing research on artificial photosynthesis.
Okay, so thank you, Mr. Chairman, for the kind introduction. And it's a bit difficult for me to give this lecture after the very inspiring lecture given by Professor Leonard, but I will do my best. So, as you see, I work on artificial photosynthesis, and today I will try to do a slightly different lecture compared to a standard scientific one. So, due to the very different backgrounds of the audience, I will try to keep the scientific, the technical part very simple as possible, even though I will try to avoid to become too trivial, so to be only like high school level. And I will try, let's say, to stay on the two sides, so both scientific, technical one, but also easy to understand. It's not easy, it's a difficult task, but I will do my best also there. So, why talking about photosynthesis? Well, I think you saw already several times this sentence, this quote from the Genesis. And actually, if you think that the thousands of biochemical reactions that we have in our body and everywhere in living systems, in my opinion, the photosynthesis is probably the most crucial one. So if you reflect about what happens in the green plants and in the bacteria, actually, the life on the earth depends on light. So the absorption of light and how the plants and the bacteria can use it, it basically keeps the systems, everything working. So it transforms the energy into something that can be used also from more complex organisms and animals and us eventually. So actually, it's not the case if the light, the creation of light is at the beginning of the Genesis. So it's just the first source of everything, and after that there is life as we know it. So how can we describe the photosynthesis? Well, this one is a probably high school level description, is the most basic one. So you have a cartoon of a plant, you have carbon dioxide going in, water going in, a bit of sunshine, and then it produces oxygen and sugars. So this is very simple. We can draw it without using chemical structures as this. So you have CO2, carbon dioxide, water, light, sugar, and oxygen. That's it. This is photosynthesis. It's very easy. Now, if you think of this, and if you think of what happens in our bodies and in all other animals except for plants, actually, when we breathe, the respiration is exactly the other way around. So we take oxygen from the air, we take sugar, or for example, when you eat like a piece of salad, And by breathing, we produce carbon dioxide, water, when it's wet, especially here in Rome, it's quite easy in summer, and some energy that partly we use, partly we dissipate in the environment as heat. So if you look, the two reactions are very connected. So basically, we use what photosynthesis produces, and we produce what photosynthesis uses, except for one point, which is this guy, the light energy. So the system doesn't violate thermodynamics. It's not a perfectly closed system because there is somebody who pays the bill, and somebody who pays the bill is actually the sun, and he pays a huge bill because imagine the enormous amount of energy that comes on the Earth every second. It's a lot, and it's a huge amount of energy, and the plants use a small amount of this to sustain all the story, right? So, okay, as I said, it's very simple. What can we say on photosynthesis? Well, if we try to scratch a little bit under the first level, let's say, you will see that the situation is a little bit more complicated. And here, I will take one moment to let you appreciate this quote by Marie Curie. I will drink a bit of water to help the synthesis in me. So basically, this sentence by Marie Curie is, I love it because it's so true. I mean, when we are in the lab, we become like children. So we are excited, we want to understand what is going on, and this is actually what pushes us even towards like when we have black days, negative results, something doesn't work, we don't know, we don't understand, and we want to understand, we want to overcome the problem. So basically, we place ourselves in front of the nature, in front of the lab problems as children, and we try to understand. And when we try to do this with the photosynthesis, what we find out is something so amazing that you say, oh, wow, it's so complicated, and you saw two reactions, it's simple. No, it's not simple at all. So let's take a closer look at the photosynthesis. We start with the main actor, which are the cell of the plants, for instance. So if you take a small slice of a leaf and you put under a normal microscope, an optical microscope, you will see the cell wall and you will see these small green dots. So these green dots actually, they look more like beads, and they're called chloroplasts. And chloroplasts are the power plants of the leaf. So at the systems where the photosynthesis takes place. And here you see the structure of one chloroplast. You see this bean-like shape. Inside you have the so-called thylakoids, which are like the green disc, which make the chloroplast green. You will see why they're green. And then there is outside a kind of a matrix, which is called stroma. And this is basically the external part of the chloroplast itself, okay? And then everything is surrounded by the membrane. Now, let's take a look at these thylakoids, and we see that actually you have, if you look at the mechanism of the photosynthesis still from far, you see that in your bean, you have some light reaction and some light-independent reaction, the so-called dark reactions of photosynthesis. So the light reactions are taking place into the thylakoid, and the dark reactions take place outside of the thylakoid, okay? So in the stroma. And you see that the process is separated. So you have on one side water coming in and oxygen and light. And on the other side, you have the CO2 and the sugar. So the system is separated. It's not, as I told before, that you have the entire reaction taking place in one step. It's not like that. And if you look even closer at the light reaction, and now I will explain in a second why we are focusing on the light reaction, because we want to copy, we want to mimic the light reaction, we want to use the energy of sun. You see that also in this case, it's not just water plus light becomes oxygen and some highly energetic molecules, but it's very, very complex. So what I'm trying to do now in the next, I hope, 5 or 10 minutes, in the first part of the talk, I will give you a more or less detailed description of this picture. So I will tell you what is going on in the light-dependent reaction. And then in the second part of the talk, I will describe how we try to mimic this to produce something useful for us. So not sugar, not highly energetic molecule, but useful for us, like hydrogen. Okay, so the beginning of everything is the absorption of light. So how light comes on the leaf? well, you have the sun, and you need something to absorb it, okay? So what is responsible for light absorption are these nice green structures, which are called light harvesting or antenna complex. So this light harvesting or antenna complex are basically a symbol of different light absorbers, which are embedded in a protein matrix. So here, those of you who are more or less familiar with biology or with chemistry, you can recognize some in these ribbons are some like proteins, and the colored structure, the colored units are like chlorophyll and other pigments, which are capable of absorbing light. And if you see we are capable of isolating the different components, and you see that they are chlorophyll, they are a series of different molecules with several double bonds. And at the end of the day, there is a final chlorophyll, which is called chlorophyll A, which is here and here at the beginning of the photosystems. So how does the energy come from here to here is via energy transfer. So this process looks a little bit like when you play the guitar, you have one string, you put one of the strings in vibration, and another one starts resonating, so starts vibrating as well, even though you didn't touch it. So that one is the resonance phenomenon, and this is exactly what is going on here. So the energy is resonating from one pigment, so either chlorophyll B or lutein or the other ones, beta carotenes, this one I think everybody heard, more or less, till chlorophyll A. And the system is so well organized that actually it managed to collect most of the visible light, except for this region, and this is the reason why chlorophyll, by the leaf look green, it is capable of collecting most of the visible light and converting all this energy and exciting the chlorophyll A. So the chlorophyll A takes all this energy and uses it for the next step, for the next reactions. Now, there is another reason why there are all these components and not only chlorophyll, and the reason is that the plant itself cannot use an umbrella to protect against the excess of light. So when the sunlight is too strong, when there is too much radiation, it should use something to protect, because too much light means photodegradation, so basically the chlorophyll is falling apart and is destroying it. So what happens is that these systems can also absorb more light, they can change slightly the structure, and so they basically prevent the photodamaging of the chlorophyll. So it's a kind of protection. They have a double goal. So as a funnel of energy and protection in case of too strong irradiation. So the first step, we have we start from here, from the photosystem 2, and you see there is this zeta scheme which I'm going to show now. So this one here, you have the energy level, and you will see that you have the two excitations, so with light, and then there are these cascades where the electrons are transferred. Okay, so we have at the end, our excited chlorophyll. So we are here at the beginning of the process. And what happens is that in photosystem 2, the light is converted into electrons. So before we had this energy transfer resonance mechanism, now we change and we use single electrons. So the electrons are actually transferred quite quickly across this system. So here you have the chlorophyll, here you have a system which is called pheophytin, and this basically a chlorophyll without the metal. The chlorophyll has a magnesium in the core. Then you have some organic structure like ketones. So you have all this chain of electrons. And at the end, you see here, I hope it's a bit small, basically the speed becomes slower and slower. So the first electron transfer is super fast. It's 3 picoseconds. Picosecond is 10 to the minus 12. It's extremely fast. Why? Because this one you can imagine like a bunny. He has to run as fast as possible, because otherwise the electron can go back. Because this one is an excited state, so it's something that is not stable. It's not supposed to survive for a long period of time. Okay, so if the bunny doesn't run fast enough, basically there is something that just catches it, and in this case it's the bunny itself that wants to go back to a ground state, which is more stable and more, let's say, thermodynamically useful, but not for the plant. So basically what happens, you have these electron transfers which become slower and slower because they depend on distance, And let's say the higher the distance, the more difficult is the back electron transfer. So this is one of the tricks. So the first one is extremely rapid, and then it can take a little bit more time. So at the end of the process, the plant produce this reduced plastoquinone, which is basically a molecule that is capable of being oxidized or reduced, so can take or give electrons quite easily, and which is used in the green plant for transferring the electron. The electron alone have to be active to go around. Okay? Then what happens when you produce this? That here at the beginning, you have a positive charge because the electrons are flowing from here to here. So what happens is that you need something to compensate this positive charge. What can you use? Well, you need something that sacrifices itself and giving electron, and this is water. So why do you produce oxygen in the photosynthesis? Because the plant doesn't need, well, it will need oxygen when there is no light, but it's a side product. It's like the CO2 for us when we make a barbecue. So for the plant, it's not useful in the moment of the photosynthesis. But it is useful because here it needs to neutralize the positive charge after the electron transfer, otherwise the system becomes charged and it cannot work anymore. That is the reason. So to oxidize water, nature has, oh my God, has a very nice system made of manganese, which is highly efficient and oxidizes water and produces oxygen. So now I go a bit faster. So in the cytochrome, basically this reduced plastoquinone gives out the electron and the protons, and the electrons are then used to oxidize, to reduce this plastocyanin, which is a protein which flows towards the second photosystem. Okay, here I'm going fast, otherwise I will talk about the rest. So in the second photosystem, you have basically the same story, so absorption of light, electron transfer, and you produce this another charge carrier, which is called ferrodoxine. So in this case, we have iron. You see, we have iron, copper. We have something that is used easy to oxidize and to reduce. Okay, at the end of the day, we make two highly energetic molecules, NADPH and ATP. So this is the photosynthesis. This is the light part of the photosynthesis. So where is the sugar? Because we say photosynthesis makes sugar. Well, the sugar is produced in the dark reactions, which don't take place during night, but just don't need light, and are making in the so-called Kelvin cycle. Okay, so in the Kelvin cycle, you produce a piece, which is then used to make sugar. So everything is quite complex. Okay, artificial photosynthesis. How do you do this in the lab? So usually when I give courses about artificial photosynthesis, I show the normal one, and I say, okay, now we like you do. So it's very difficult. We don't want, of course, to make exactly the same. We want to modify a little bit to use for our goals. And our goals is to make hydrogen, because it's something green and it's something that we can use to, for example, for cars as a fuel, okay? So the way is, you think in a natural way, but you try to go synthetic. So the glossary is, what do you need for artificial photosynthesis? Well, first of all, you need something to absorb light, you need something to oxidize water, this we need as well, and you need something to produce hydrogen, which is our goal, okay? And more important, we need something to put all together and to make it stable and working. And this is one of the big challenges. So the oldest topics in artificial photosynthesis is the study of light harvesting. So we need something stable, we need short distances, very well organized. But this we know how to do, okay? It's the easiest way of studying, of approaching artificial photosynthesis. So in this case, for example, you have a porphyrin and Fullerene system. If you excite here, you have a transfer of energy, the dark vibrating quite fast. Then you have another transfer of energy, and then at the end, you have an electron transfer. So you create a positively and negatively charged unit. And then you have the back electron transfer. So in 1 nanosecond, the system goes down and is dissipated the energy. Well, the coupling is efficient, but the reaction center doesn't work. This one doesn't perform any reaction that we can use. So what do we need? We need something to absorb light and the catalysts, which are capable of performing reaction, and we need the right potential. So everything should be well organized to have these electron flows with the right time, with the right kinetic, and And we need something that can make reactions. So the first idea is to use multi-component, just mix all together in the solution and go. Okay, so in this case, you have something that absorbs light here, the reducing agent, and you need some sacrificial donors. This one works a little bit. It doesn't take so much time to assemble, but you have no control. So it's the system itself that decides, the kinetic that decides everything. So a better way is to use a so-called supramolecular system where you have everything connected except the sacrificial species. And in this case, you can have a good control over distance. So in the kinetic, you can really tune a little bit, but the synthesis is more demanding. So you have to work a little bit more. So which kind of catalyst can we use? These are examples of reduction and oxidation catalysts. The problem of this is that the reduction catalysts, they tend to fall apart, so we need excess of ligands. And the oxidation catalysts, we are not unfortunately as good as nature, they tend to oxidize themselves. So except for this one, which is based only on inorganic systems, the other one have tendency to get oxidized, which is not good. And this is a big part of the work on artificial photosynthesis nowadays. So some nutrients, the semiconductor-based systems are quite recent and they are very, very stable. So in this case, you have semiconductor nanoparticles connected to a reducing agent, sacrificial species as well, I will not finish, and they work well, okay? But you need sacrificial species. Another nutrient is tandem cell. This one is really imitates what happens in nature. You have two sensitizers and you can overcome more or less the problem of the potentials. But they still use expensive components like platinum. And now the very last slide, I was more or less in time, are integrated systems. So these one are so far the only two systems that are capable of splitting water with light. One is the so-called artificial lift. So you have this kind of silicon-based system with two catalysts. This one is the picture. You irradiate and you make oxygen and hydrogen. What is the problem? That the architecture is very, very expensive. So this one they tried to make like for an industrial production and they gave up because it was too expensive to make. And another one, the last one, is this one. So same story, separated. This one works better, but the yield is too low. So in conclusion, we try to make hydrogen because we need it. We need components and architecture which are cheap and efficient, so we can sacrifice the cost. It works very well. If it doesn't work so much, we have to stay on a low cost. And so far, they use sacrificial species, which are not useful. So you just move the problem of who pays the bill, and we don't need it. So let me conclude with a picture of Fribourg. Let me thank you for your attention. I ran out of time. Thank you.
Thank you. We have time, I think, for one question.
You said that photosynthesis produces oxygen, among other things, and oxygen, of course, is important for our life. How much of the oxygen in the atmosphere is actually produced by photosynthesis? I'm not an expert, but I've heard that there are some bacteria at the bottom of the ocean which produce most of it.
This is a very good question. I have no idea, to be honest. I think most of the oxygen, the molecular one, is produced by photosynthesis. Because, I mean, even the plants uses oxygen to breathe. We never think of that, but during night, the plants breathe exactly as us. So I think most of the oxygen that we see in the air actually comes from this cycle. So it just transforms starting from water.
Thank you, Dr. Rudy. I wish you every success in overcoming the aspirate here, because if you succeed, it will make a huge difference. Our next speaker is Professor Marco Personelli. He's going to talk to us about the first light in the universe. He is a full professor in astronomy and astrophysics at the University of Milano, director of the PhD School of Physics, and one of the leading scientists of the Planck space mission of the European Space Agency.
Thank you very much. Thank you for this invitation to such a stimulating meeting here in Rome. And yes, so my contribution will be to speak about light as we receive it from far away. And clearly, light in general, but I would say especially light that we receive from the stars, has a deep symbolic dimension. And clearly, every ancient civilization, the artistic expression of every culture has seen in light something really crucial. Now, as physicists, as astrophysicists, we use light as a messenger from far away. And this is just a vision of the sky as we could see it if we happen to be in a very dark and clear night. And we have to realize, first of all, that the kind of scenery that we see with our eyes when we look at the sky is very much limited or determined by the range of wavelength which our eyes are sensitive to. So this beautiful view of these dots of light in the black background, which is the night sky, is something that is peculiar to the kind of reception that we are able to use. And in fact, in this image, for example, some of you would recognize the Orion constellation, for example, here. Well, if our eyes were sensitive to infrared, for example, the scenery would be completely different. We would see clouds of dark, which appears dark to us, but is essentially dust and gas interstellar medium. So, if we take the entire energy that light in the universe as a whole is producing in the optical, in the visible range, then we can reconstruct a certain spectrum, a certain distribution here, but then we can span through the wavelength range all over, and we can realize that infrared, for example, contributes at least as much as the optical, and then the radio and the UV and X-ray, high energy photons here. And so we can reconstruct the more generalized concept of light as it comes from our universe. And we know that the stars are responsible for the light we can see, even from galaxies far away. And we have a good idea of which physical mechanism produce all the other kind of electromagnetic radiation throughout the spectrum. The other thing that I want to mention at the beginning is the velocity of light. Light is extremely fast, but not infinitely fast. And that has a deep consequence in the way we can look at the universe. 300,000 kilometers per second is fast enough that we can treat light as instantaneous propagator in human, let's say, normal distances. But as we look at the sky, precisely the sky we can see with naked eye, for example, Sirius, this very bright star here, the light from this star took about 8.5 years to reach us. And if we look at this star here, which is one of the most luminous stars in the visible sky, it took 1,300 years to reach us. We go back to the a millennium and more ago. And if we look at even the closest galaxy, this is a system that includes something like 100 billion stars. This is the Andromeda galaxy. Well, this light traveled for 2.5 million years before reaching us. So we are looking at these objects as they were in the past. We go back in the past, the more we look at sources that are far away from us. This is very important for our work as cosmologists because we can have an idea of the history of the universe and not only about the structure of the universe in the local environment. Okay, so that means let's look how far we can go with our observations and with this animation. This is taking us with the Hubble Space Telescope more or less in this region where we have the deepest image so far of the universe of galaxies. The universe is full of the structures we call galaxies, and they are, as far as billion years from light years from us. So every point you can see here now in this animation is a galaxy, and now we are going to the deepest image, which is called the Hubble Ultra Deep Field. And this field here contains something like 10,000 galaxies in a very small region of the sky. And what is important to notice is that there are some of these almost invisible sources here. They are the most distant galaxies that we know. And the light from these galaxies took something like 13 billion years to reach us. So now you have to answer, you see, the question is, how does the universe as a whole behave in the billion-year timescale? And today we know that the universe is changing as a whole because it is expanding. So, and we can, we can calculate from the rate of the expansion that about 14 billion years ago, any 2 points in the universe were very close to each other. The universe was much smaller and much hotter than it is today. And that was about 14 billion years ago. So you see, these far galaxies belong to a very young universe, only 1 billion years old. And therefore, the question is there a light that comes from even further away, such that we can get information directly from a universe that was even closer to the beginning of the expansion? And the answer is yes. And amazingly, the answer is yes, we can. There is such a light. And this light comes really from the black background that we see as a background of any picture like this. This was discovered exactly 50 years ago by Bentley and Wilson, who got the Nobel Prize for this. And this is called the cosmic microwave background. This is because the peak of this radiation is in the microwave. This light is a few millimeters wavelength. And it comes almost uniformly from every region in the sky. So if we could draw just a sketch of the universe as a whole, as we can see it with our eyes and with our instruments, this is how it looks like. We are not physically or geographically at the center of the universe, but because the universe is uniform, anywhere an observer is in the universe, he would see a spherical symmetry around him in space and time, right? So we see galaxies that are further and further away, and we go closer and closer to the beginning of the universe, of the expansion of the universe. The temperature increases to the point that somewhere here, we get a universe that is completely ionized and opaque to light. And this is the region of space-time from which we receive this background light, the cosmic microwave background that reaches us from every region in the sky. Now, our business, our research is really to study this first light in the universe with great detail. And with that, we can learn about things that really happen also in the very first phase of the existence of the universe. This light we received when the universe was about 380,000 years old, so that is about 0.003% of the current age of the universe. But then we can infer through physics, what happened, at least to some extent, what happened in the very early and hot phase of the universe. But now let's go back to this plot. All of this radiation that is represented here in this broad spectrum is produced by sources in the universe. You see, whereas the cosmic microwave background is really light, it is originally into the universe before any source was produced. And if we plot that light in this plot, you see that the amount of energy that is connected to this early light overwhelms the light of any other source in the universe. 99% of the radiation energy now in the universe is coming from the first light of the universe, from the black background. Everything else is a detail in terms of energy. Now, we know today much more than Penzias and Wilson about this light. This map is the first full sky map of the intensity of the cosmic microwave background. It was produced by this satellite, COBE, in 1992, and John Mather and George Smoot again got the Nobel Prize for this work. And this is a full sky map, okay? So this is just a representation of the celestial sphere, and the points, the regions that are red and blue are really distinguished by a very tiny difference in the intensity that we measure in terms of temperature, okay? But the discovery of this anisotropy, these fluctuations in the intensity of the early light was very important because it was expected from theory, and it comes from the fact that we believe from physics that even in those early times, the universe needed to have some irregularities due to gravitational, let's say, seeds of the structure that we see in the current universe. So in other words, what we can see here is the effect of region in that early hot universe where the material was more or less dense and with different movement, and the structure was starting to be generated. Now, you see, the angular resolution in this image is very poor, in fact. It's only 7 degrees. So the smaller detail you can see in this map is about 7 degrees. And the fact is that most of the information about the early universe, about the structure and evolution of the universe that this early light can give us, comes from more detailed analysis of this radiation. And let me just sketch the reason why that is. This is a representation of the statistics of these fluctuations. This was known by the time the COBE results were produced, and this is a spectrum of the anisotropy, of the intensity of the anisotropy as a function of the angular scale. And so if we go to large angular scales, such as the map that we just saw, in fact, all the red points are only here, we are only at the beginning of this power spectrum, which has most of the information, these peaks and valleys, at smaller angular scale. In other words, we need to have a more high definition image of that early light in order to learn about the details of the statistics of the matter and radiation in the early universe. And why it is so interesting to actually measure the details of this curve here, this power spectrum, because the details of these peaks and valleys depend on the kind of medium in which this structure formation was taking place. So if we can actually measure that, we know we can learn, for example, about the composition of the universe, how much baryonic matter is there in the universe, how much dark matter or other form of energy and matter are there in the universe. We can learn that by measuring this in great detail. And also we can measure very accurately the rate of expansion of the universe and the geometry overall in the universe. So this is really an important objective. And that was the objective of the Planck mission, in which I've been working for the past 23 years or so. Okay, and you can see how the variation of some of the parameters change the details of the spectrum. And so clearly a measure of that would be quite informative. So here is the satellite, which is built to look back to the dawn of time, really. And this is how the satellite has been getting a deep image of this early light in the universe. It is spinning on its axis, 1 RPM. And so by following the Earth, every 6 months is producing a full sky image of the early light, of the cosmic microwave background. And you will see here this scanning strategy And you see these fluctuations at high resolution. This is just an animation, but I will show the data to you in a moment. And these fluctuations really represent the universe that is taking form in the very early stage of its expansion. This very bright red bar that you can see here is essentially our galaxy, because we can go outside of the atmosphere not to get the confusion from the atmosphere, but obviously we are within our galaxy. These are galactic coordinates, and the galaxy does emit radiation which pollutes to some extent the image that we really are interested in, which is the rest here. And so this is not an animation. These are the real data from Planck. We measured 9 different frequency bands in different colors, precisely to be able to discriminate light that comes from the bottom of the universe and light that comes from our galaxy. And by making a careful analysis of these 9 maps, we can get this, which is the most accurate image of the early universe that has ever been produced. And you can see, compared to the previous image, the level of detail that we get here. Now, the objective was to get this curve with high precision, and you can see here the amazing, I would say really amazing coherence of the data with this model, which is only 6 parameters model, from which we can get the amount of baryonic matter, dark energy, the constituents of the universe. And today, light is only 0.01% of the content, the energy content of the universe. But we know how matter, dark energy here, which is something like 70%, and also radiation, light, behave in cosmic history. So if we extrapolate back in the past, we see that if we go sufficiently in early time, radiation was actually more important than matter in dominating the energy of the universe. So there are 3 eras. We are now in the dark energy era. There's 70% of dark energy today, and matter era that goes back to the first 50,000 years. But before then, in the first phase of the universe, really radiation was dominating. And so, in fact, if we go back to the first year after the Big Bang, you will find out that the universe was essentially pure light. And this is because light has a different just very quickly, because time is over, we are now also able to analyze the Planck data in polarization. This is another way to characterize the information from light. This is our galaxy. This is the primordial light in the microwave background. And from that, we are able also to see the hints from the generation of the first stars, which have a peculiar signature in the polarization of the microwave background. So I guess I should conclude now. And so just to try to make some of the points I was trying to contribute, it is really, I think, a privilege for us as human beings to be able to see something that happened really close to the beginning of the history of the universe. This is far from obvious. Even the location we are in our galaxy is something that is important for this possibility to be there, okay? So we have this privilege. And we have seen that light from the beginning of the universe, the cosmic microwave backgrounds, is the dominant form of radiation, of light, if you like, in the present universe. But light as a whole in the present universe is only 0.01% of the total energy budget. However, if we go back in the beginning of the universe, light was the dominant energy in the universe as a whole. It's enough to go back in the first millennia, let's say, after the Big Bang, which is a very, very tiny time compared to the 14 billion years that it is today. We have seen also that technology really allows us to probe space and time with light that really travels from the outer boundaries of the visible universe to us. And we can do that with great precision. This is incredible too. I mean, this is really something that is not often seen in physics. And the final remark is that light, as I said in the beginning, is really a very deep symbolic value. And all religions, for example, the religious sense of human beings has been formed in the idea of light. What I would like to say is that, in my view at least, these new findings that cosmology is bringing to us through science do not hinder the unique symbolic dimension of light. On the opposite, they provide, I think, new and unexpected aesthetic and symbolic power. Thank you.
I think we have time for a couple of questions, and I'm going to take Chairman's privilege and ask the first question, which is, what are the chances that we will see a ESA or NASA mission or satellite named after Father Lemaitre, who came up with the Big Bang theory in the first place?
It's actually a very good and appropriate question, because obviously now Planck is still on the scenery, and however, there are already studies for, at least on the ESA side, for a post-Planck space mission, looking at polarization in particular to high precision. And I have to say, I can say, although it's not a public or official thing, that there is quite a large consensus among our community around the name of Lemaitre for such a future mission. It has been discussed, and I think most of the people are in agreement with that.
Hello. Now, you have explained so many complicated things connected to the astrophysics in a very simple way. I'm here.
Oh, okay.
Anyway, thank you so much, because we have got a clear picture of what is happening over here. But now I have got a simple doubt. Does that CMB pattern mean that our universe has got some boundary? And if so, beyond that boundary, what is that?
You see, the point is that I think that the best way to try you see, the point is that we have all evidence is telling us that the universe as we see it today is emerging from this very hot and very dense initial state, okay? Now, this initial state, we call initial state because we see all the development starting from these initial conditions. Now, time is the key. We can date with Planck, we can measure with 0.5% accuracy the date of birth of the universe, which is 13.81 billion years ago, okay? So now, if we go in the first microseconds or even more, the first very tiny fraction of a second, you see, we can speculate today about what happened at 10 to the minus 35 seconds. This is the idea of inflation that might have shaped the structure of geometry, for example, in the universe. But we can go as far as this very tiny fraction of a second after the Big Bang. But to some extent, your question is like asking what was before. And so we go back to St. Augustine that we saw before, right? And the concept of time becomes into question very, very deeply. And in my view, we as scientists, we should be humble and very, let's say, courageous at the same time, but always speak about things that we can speak about.
this could be a very interesting discussion, but I really have to move the conference along, so thank you very much, Mr. President. Michael Berry, you wish to speak, and we have a number of people who wish to speak. We have to make.
Sorry, I thought you were truncating the discussion.
I am trying to truncate the discussion, but
Well, I wonder what you think of this idea. It doesn't seem very sensible to use the same scale of time for things that happened long ago when the universe was much hotter. If instead you measure time according to the number of things that can happen in any unit, this was much faster in the past, then we would replace time by a logarithmic measure in which, in this sensible scale of rate at which things happened, what we call the Big Bang is actually minus infinity. And this seems much more sensible. And then you would have no question about what happened before, because infinitely many things will have happened as we go farther back. Now, I'm sure this is an idea that is discussed in cosmology, and I would like to hear your views on it.
Right. So, well, I think there are two ways to, as far as I can try to follow your thought, there are two ways to take the point. One is a matter of representation. It's a very convenient way to represent time in logarithmic scale, and that is what is usually done, right? It makes a lot of sense. It allows to see things, and even this is a sort of logarithmic thing, because obviously you compare thousands of years with nanoseconds or stuff like that. That's a very good way. But the second, more deep question is there a physical meaning attached to that? Yes. And the point here is that so far, if you take time linearly, physics seems to work very well, at least with experimental evidence. It goes very well, at least for the creation of the first light elements, the nucleosynthesis, the cosmological nucleosynthesis, which is a few minutes after the Big Bang. So if you then ask us to stretch time in a strange way, then we don't understand physics anymore.
Oh, yeah, I'm sorry, I don't understand that. There's no conflict in physics at all. Well, it'd be just the same if you replace t by e to the t or log t. It's just what's sensible is that you're measuring time in units of the rate at which things happen when the universe is much hotter.
Right. That's right. Then it is really a matter of representation. And so it's like we could do that with our own age, right? For example, things happen very much faster with my nephew that is younger than.
Yes, and that's very sensible.
Yeah.
thank you. Before introducing the next distinguished speaker, I'd like to make a brief personal statement, because one of the upcoming speakers actually mentioned to me yesterday that he thought there wasn't any real point in having science try to talk to religion, that they both should continue to go their separate ways. But I do know that in the United States they are entangled through politics, and we have a very dangerous, actually, division between science and religion. And with some of the upcoming questions, such as equitable, sustainable energy for everyone on this planet, genomics, ethics, and so on, the list is very, very long. There is real reason for dialogue between science and religion. And I think it's because of that I really like, this also, by the way, meets one of the requirements of the United Nations in declaring the International Year of Life, that all aspects be involved. So thanks to Eugenio Fazio, who's our next speaker, and Father Pasquale for arranging this meeting, which I think is actually deeper than perhaps many of us realized at the time. Our next speaker is Eugenio Fazio. He's going to talk to us about biomorphic optical functions. He graduated from the University of Roma La Sapienza, and did postdoctoral studies in the desert in Tucson at the Optical Sciences Center, at the Ecole Polytechnique, and at the Laser Research Center in Vilnius University in Lithuania, and at the University Polytechnic de Catalunya in Barcelona. So he's got a multicultural background here. He's professor of experimental physics at La Sapienza University, specializing as an expert in optical nonlinear dynamics in bulk materials, waveguides, and nanostructured media. And he has published more than 130 scientific papers. His recent interest is moving towards biophotonics. Eugenio.
Thank you very much, Eugene. As I said already yesterday, thank you very much for being here. I have to ask Albert Ruggi to excuse me because I will speak a little bit about chlorophyll and I don't know anything about that. So I'll try. When I was kids, the teacher at the school asked me to make the usual experiment to put beans on the cotton and to see them germinating. So I went back home and then asked my mother, please buy beans because tomorrow I have to make the experiment. Then I went back the day after and I asked Mama, where are my beans? And then said, wait a moment. And she took dry beans and I said, wait a moment, they are dead. I need fresh one, I need green one. And said, no, use them. Said, no, they are dead. No, don't worry, you use them, trust me. I don't trust you. Then I was obliged, and after a few days, I saw them green, and I was astonished. I was astonished a second time, a few years ago, that a colleague came to me and said, do you know that beans, when start germinating, they produce light? Light? How is possible? And this light is composed by coherent photon. Coherent photon, no, that's not possible. It's only laser do that. So in this here, together with this colleague, we have been trying to measure that light emitted by beans during their germination. Usually, this kind of beam is called biophotons or dark luminescence, ultra-weak light emission. Different way of saying the same stuff. So the internal part of the beams that is useful after, then when you open a beam, you find all this area here, which is the cotyledon, which is almost the energy of the beam. And then here you have the embryon with the plumula, which is the first leaf. We built a luminometer. It's just a photon counting system. With everything was in the dark, and you have also a beam here, which can take water without getting light. So everything is in the dark, and the box is grounded in order to avoid any electromagnetic influence from outside. So this is the signal that you have from beams, or from cotyledon, so part of the beams, when you put water on them. So at the beginning you have just a noise background, then you put water, and then you see that optical signal I'm measuring in the optical band. The optical signal just jump up and then it goes down. And then different experiments show always the same behavior with almost the same time constant. Then I wanted to understand if this was something typical of living beings. And then we denaturate the beans. So we put the beans in the oven and we kill them. And actually we observed also a signal from dead beans. They were really dead. But from dead beans, the dynamics in time is different. because you have a first decay and then a very slow one. Then try to understand this. Mr. Pop and Mr. Lee, something like 20 years ago, demonstrated that hyperbolic relaxation is a sufficient condition for a fully coherent ergodic field. Then when you have an ergodic, a coherent ergodic system, the decay is not anymore exponential, it became hyperbolic. And then if you see in a log scale, the signal from denaturated cotyledons, you have here the noise. The blue line that you have here is just exactly the same system which was smoothed in order to take out the noise. So you have a background, you give water, and this is jumping up. You have a first decay, you have a second decay, and this is just almost the shock noise of the detector. So you get imbibition, you get the first exponential decay, but after a while, hyperbolic decay can. So for denaturated beings, you have molecules which are oscillators, which are almost independent of each other, but after a while, they feel each other and they start to have coherent dynamics. While with the not denaturated, so natural beings, you don't have these both dynamics. So you have inhibition and you have just one exponential decay. So coherence seems to come only for free oscillators, but if the oscillators are somehow connected biologically, then they are not able anymore to induce coherent dynamics and they are independent. If you analyze not part of the beam, but the entire beam, how does it look like? So you have this a small spike here is just when you open the luminometer, so it's not interfere with dynamics. Then here you give water, you have first relaxation, water enter inside the codes. And then you have a big spike of light, which is characteristic of the entrance of the water inside the beam. Then the second signal that we're not able completely to understand. But then if you look at very long time scale, which is almost 3 days, so you have the entrance of the water, the second spike, and then the germination. And when the system starts germinating, it's just like a lamp. It's emitting light everywhere. What is the light for? Some of the questions of this conference started from this measurement that I did. I mean, that's useful. The light is useful for the beings, or is it just a waste? I don't think so. So I was trying to understand how this light works. And I come from laser science. So when I heard about coherent photon, I started saying, this is a resonator, it's an optical resonator. So I start analyzing beams as optical resonators. So you see this shape, that's a real shape from a cannellino beam. Our friends from Florence knows well cannellinos because it's a good recipes for cooking, for kitchen. Yeah. So that's a laser scan of a cannellino that you see that it's a typical shape. It looks like mirrors, like conversion mirrors here. Then we started to analyze beams from geometrical point of view. And you see that if you analyze dimension, and you see that they are in a special arrangement one each other. So it's not, the shape is not casual, as we believe from the light, from the nature. Then at the beginning, I thought there are just 2 mirrors, actually 3 mirrors, because there's a small mirror here, a big mirror here, a big mirror here. Even if the upper part and the lower part looks like perfect round surfaces. So I was thinking just light which was going back and forth. Someone of you could ask me how light could pass inside the beam because it's not empty. Yes, it's true, it's not empty, but you know that you have 2 cotyledons which are separated and then space in between. And actually, the embryo is exactly in between the 2 cotyledons. So that's enough space to let the light passing through. If you analyze the center of curvature of the whole shape, you realize that the beam is not symmetrical. That's the symmetric line, and the center of curvature is going up. That's a big difference. You see that all this shape is somehow modified in one direction. And this is, again, not casual. Actually, this is the importance of the beam as a resonator. And that's all the geometry of the beam that we analyzed. And you see how this is not symmetrical. Then to see how this it was working optically, from this shape, which is a natural one, we took a shape using a basis spline. Then we simulate possibility to have a light source inside the beam. And we did the analysis of ray tracing, like a beam in a reflecting mirror. So light which was emitted by these sources arrived to the coat and then is reflected back and forth. And actually, this is what you obtain from the ray tracing. This is an artificial symmetric shape that we did by computer. And this is the real shape. So the symmetric shape, you have here, of course, a lot of light, but here is where we put our light source. Then you have this part here, and you can recognize that light is traveling through the coat, arrives here, and then is focused back here. And you get exactly the same here. But actually, a real beam is not symmetrical, it's just twisted in one direction. And this twist is exactly in the direction where the embryo is. So this asymmetry in the beam shape make that this focus here is just losing power, while the other focus is getting more and more power. So if you make a comparison of what we have seen or the simulation with the real shape, you discover that this focus is exactly in the position of the plumula of the first leaf of the embryo. So this was the third time I was astonished by beads. So is this true? I mean, it's just a numerical simulation. How can I realize this? And then I wanted to use chlorophyll. And then I make some bean germinating in the dark. These beans were kept in 3 dark boxes in a dark lantern. It was dark. And then when you take out the embryo, they are green. And say, yeah, okay, so the bean is able to produce chlorophyll. And then I went to some colleague from biology and he said, no, you don't know yet, because this can be chlorophyll, this can be chlorophyllidis. It's a precursor. It's something that is arrived before the chlorophyll, which is always green, green color. You have to make a real precise numerical analysis in spectroscopy, and I had no chance for that. So how to discriminate this? And then I went back to my knowledge in optics, and we did endoscopy of beams. So we put a whole optical fiber, and we went inside, and we did photon counting inside the beam. And here you have the O, here you have the embryo, and just making this measurement with almost 100 beams, we were able to make a mapping inside the beam. Actually, it's very important the direction of your optical fiber, because if optical fiber is not coupled with this focus, light will not enter inside the fiber. So we went exactly in this direction, which actually is tangent on the geometric position of the center of the curvatures, which is almost 2F lenses. And this is the experiment. So here is the theory was the numerics arrived, and this is the experiment. And you see that where you have the focus, you get a lot of light. Here should be very bright, here is darker, but you have a shadowing from the plumula. So plumula is getting a lot of light, and you arrive from below, and then you don't see this light because it's kept by the plumula. But if you go a little bit lower and a little bit upper from the plumula, you don't see much. So in conclusion, we measure biophoton emission from the entire seed and from part of them. And the seed and part of them has proper dynamics. Coherent statistics seems to be present only for denaturated matter. Seed indeed act like optical resonator, and it seems that there is electromagnetic signaling within the seed. And as I said even yesterday, there are 3 reasons because the signaling is occurring. One is to trigger the germination start. The second is to inform the embryo that the seed is getting water. And third, provide light energy for chlorophyll synthesis. Could we speculate, this is just a pure speculation, could we speculate that electromagnetic signaling occur really, generally in nature? I don't know, really, I cannot tell you. But there are a lot of similarity in shapes, because red beans looks very much like kidneys. Actually, you don't recognize them if you don't know what they are. Broad beans, in Italian fave, who is from south knows about the puree di fave, which is very good, but looks very much like lambs. But what is more astonishing is chicken peas. Do you recognize brains? They are not different. And even mitochondria, I'm sorry, I've forgotten H there, looks very much like antennas. So does exist electromagnetic signal in nature? I don't know. But many shapes of nature say that. Thank you very much.
So we have time for 1 or 2 questions.
Maybe just if you would make a small clarification. Of course, inside the bean, it's scattering media. I mean, you can't do any race tracing because everything is scattered. But you do all this in the slit in between the 2 halves, where there is free light propagation, not everywhere, because there everything is blurred by scattering, of course.
Absolutely, yes. And if you look at the beans after you let him to get in the water, you discover that the space in between cotyledon is getting larger and larger. So there's always more and more space free for the light to travel inside.
Well, it's astonishing talk. My question was asked, I'm sorry.
Professor Fazio, your lecture of beam physics was fascinating. Beam physics from beam optics, I don't know. My question is that you observe differences between, let's say, vivid and dense beams.
Yes.
What about the spectra? They also differ wavelengths.
Yeah. Jan, you're always able to arrive to the point. Always. I know you. I know you since 30 years ago, and you always arrive to the point. We are performing now some measurements. There are different lights and different spectra arriving from different parts of the beat. Code has a special spectrum. Cotyledon has a special spectrum. We believe that the spectrum is strongly related with the information that is carrying. Code has some sensor which are able to say to the beams how much water, how much oxygen, how much CO2 is present in the environment. So to inhibit or to allow germination. Cotyledon is more the spectral emission from cotyledon is almost overlapping with the sun spectrum, because we believe that the light arriving on the plumula, it's more connected with the chlorophyll production. So it should very much looks like a solar one. So different spectra for different functionalities.
Thank you. Thank you again, Eugenio. Of course, it is from the theme that we have the word lens, possibly the association by Heisen, the famous Arabic optical scientist, but that is generally regarded as the word lens. Our next speaker is Claudio Tunis, who is a scientific consultant and former assistant director of UNESCO's International Center for Theoretical Physics in Trieste, where he presently promotes the use of advanced physics methods in human origin studies. Let me say before he starts that ICTP does not get the recognition that it really deserves in the world, and the support by the Italian government, the constant support for it is actually quite marvelous. It is doing a wonderful job of trying to bring science to the developing world, and I think that that is something that should be recognized a lot more.
Okay, thank you, Mr. Chairman, and also for the nice word you said about my institution. Yes, I'm a physicist. I'm going to talk about human origins, but as my friend and colleague Giorgio Manzi, a renowned Italian paleoanthropologist, say, he always tell me that I am a physicist linked to paleoanthropology, so fisico imprestato alla paleoantropologia. Well, I would like, first of all, to thank also the organizer, Father Pasquale, for organizing, and the other organizers of this conference for giving me the opportunity to attend this really conference, which is making my brain rotating by 360 degrees around various topics. So really, it's an exciting conference. Also, this topic, human origins and evolution, has attracted the interest of theology, philosophy for thousands of years. Now, science has been studying systematically, I would say, and seriously human origins and evolution in the last 150 years. And so my task today is really to talk mainly about the contribution of light-based methods to study human origins. Actually, this is a very famous sentence connecting light and human origins, and that is in the last pages of The Origin of a Species by Darwin, 1859. And this is the only reference in this book on the origin of species to human origins. So Darwin actually was not aware that just a little more than 30 years later, a physicist, Wilhelm Röntgen, would have discovered a special light, X-rays, that really would allow to fulfill this Darwin prophecy in many ways, as I will show you. Now, let me show you this group of extended human family, which is a reconstruction. If you go to Paris, to the place where Elizabeth Dainese is rebuilding, is creating these early humans and hominins, that really would be a good experience. And here you see the whole family reconstructed using forensic methods that the French police, I guess, is using when you find the skull to recreate the face of the person that was murdered or whatever. So these methods are used together with X-ray tomography and so on to reconstruct the faces of our ancient ancestors. And it's really quite inspiring to have this possibility. And it's a lot of work. And if you want to, actually, you can buy a Neanderthal man that you can see here at the end. And that would be like 100,000 euro if you want to have, in your house, a real Neanderthal man. So Charles Darwin then, in the next book, 1871, on the descent of man, where he discussed human origins, he said that our ancestors came from Africa, and that was only based on the similarity between us and the apes that you can find in Africa, the gorillas and the chimpanzees and so on. And Darwin at the beginning had many problems, with this kind actually of talking about the similarity between us and the apes, as you know, and so on. And again, this will be demonstrated in many, many years of work during the last 150 years. Now, so the work that scientists have to do is to find where there are the remains of human ancestors. When did they live? How was their life history? Meaning, how long was, like their childhood? How long were they living and so on? How was their brain? What were they eating? When did we develop a modern mind? And actually, it's coming out that we were not the only human species that evolved a modern mind. Maybe also the Neanderthal had their kind of modern mind. Now the answers come from what I call light science, using the full spectrum, not only the visible light. Darwin would have used just a lens with some light to look at the external structure of the specimen. But again, we can use the whole spectrum from microwaves in the ground penetrating radar to look underground and search for human remains, I will show you other examples of remote sensing, but then we can use, as I said, the X-rays also for looking inside, look at the microstructure of human remains, of ancient human remains. This is just an example about the new methods that we are using based on light in archaeology. So this is LIDAR, light detection and ranging, which now every archaeologist wants to use, because you can get information on the landscape, on the ground, getting rid of, like if there is a forest and you cannot see the structure of the land, with using the software applied to the data that you obtain with this method, which is based on laser scanning. So there are a number of pulses going every second down per every square meter, and you then can store all this information, and you can clean all the background coming from trees and other elements that you have on the ground, and get a beautiful picture that allows you to discover prehistoric sites and also other kind of interesting ancient structures. For example, in the Trieste area, with my group, we discovered in 6 months, you can see there the red spots, we doubled the number of prehistoric sites known in that part of Italy, which is Friuli Venezia Giulia, comparing to what was discovered in 100 years of hard work by the conventional archaeological methods. So that just to give you an idea. And we found also other interesting things, like we published, you can see here the publication, very recent, 2015, on the Proceeding National Academy of Sciences. We discovered the most ancient military Roman camp in Europe. Actually, it's a system of military camps that go back to 2,200 years ago. And Trieste actually evolved from these military camps, the city of Trieste. That was published recently. But just to give you an idea about the power of this technique, you see this is one area on the Trieste karst. From the helicopter you see only trees, but then when you apply the LIDAR, the laser scanning method, you can see this structure. And this is a Castelliere, is a protohistoric Bronze Age structure on the top of the hill. And this is how, you know, the archaeologists in the early 1900s were making this kind of sketches of these kind of structures. They could do that. Those days was much easier because there were no trees on the karstic area. There were no trees in the karstic area near Trieste, so you could really see this structure. Now it would not be possible because there are all these forests. But besides confirming previous sites, we found, as I said before, many new ones. So, okay, so that is just to give you an idea about the use of light to look for prehistoric sites, hominid-related sites. When did they live? That is very important. So to give you some chronology, I just give you an example of potassium-argon dating, which has been used to date most of the human remains, say from East Africa, but also from Indonesia, that is based on the radioactivity of potassium-40, which is decaying in argon-40 with a half-life of 1.2 about billion years. So you can date all the human history, going back to when we separated from the common ancestor that we had with the sheep, and that is about 6-7 million years ago. And that is predicted, for example, from the genetic analysis. And these are all the different hominids that you have, about 20 species of hominids that you have in the last 6 million years. And then after about 2.5 million years, but recently there was a paper in Nature a few weeks ago that we discovered that even before 3 million years ago, people were already making tools, and that is before the evolution of the Homo genus. So, you know, every month in practice there are new discoveries in human evolution, but there are many based again on light-related method. Why light-related? Because in the new instruments like this one in the US, laser is used to extract the gas argon from the specimen that you want to date. Just to mention another method, it's very interesting to study the last 2 million years. This is when we have the evolution of the genus Homo, originally in Africa, but then it went for the first time out of Africa to Europe and to Asia, and then you have the evolution of the first Homo erectus in Homo heidelbergensis, and this species went to Europe, to Eurasia, and evolved into the Neanderthal. Instead, in Africa, it evolved in Homo sapiens. So you can study this with other methods, a method which is really becoming very powerful, and it was recently celebrated the 30-year anniversary with this paper in Nature, is optically stimulated luminescence dating, which is becoming more precise, and it can date the last 500,000 years or maybe 1 million years, so quite beyond the radiocarbon method. But I'm mentioning this because this is a light-based method. And again, it works because in a mineral grain, you have the light of the sun, which is erasing the luminescence signal. But then when the mineral is buried underground, the natural radioactivity would accumulate luminescence. And using your laser, you can extract this luminescence signal and tell what is the age of the sediments, and hence the age of the hominid remains of stone tools that you have in those sediments. So again, these are examples. So we can reconstruct in time the different hominids that we have. Actually, it's a bush. We have several species living sometimes in the same area together at the same time. For example, recently, again, 2 weeks ago, Nature paper, a new Australopithecus species has been discovered that was living about 3.3 million years ago together with Australopithecus afarensis, Lucy. And so there again, and they were living in the same area in Afar, in East Africa. And if you go to recent time, you have 4 human species on the planet at the same time. Homo sapiens, and then the Neanderthals in Eurasia. You had the human that was recently discovered in Siberia, the Denisova, still don't have a scientific name, identified only by genetic analysis. I'm not going to tell much about genetics, but again, in genetics, the new next generation sequencers, the lasers are very important because often the sequencing is done exciting the fluorescence in the basis of the DNA and so on again, so light-based science to get to the unbelievable result that you have with the genomics of these ancient species. So we can compare them in great detail, and we can even evaluate what was the degree of interbreeding. There is interbreeding between our ancestors, direct ancestors, Homo sapiens, with the Neanderthals and with the Denisova people. That sometimes was useful, sometimes was less useful. Then, so let's move to X-rays. This is the system we are using at ICTP to doing this super tomography, X-ray tomography, which is about 1,000 times more, it has 1,000 times more resolution, space resolution than hospital CT scanning. And you can really get, for example, this is the mandible of a Neanderthal child from Italy, from southern Italy 100,000 years ago. And you can see that using then the data from the micro-CT, you can get a bone transparent and look at all the teeth inside the mandible of this Neanderthal child, also those that are not out yet. And again, you can study the life history of and then you can also then use a synchrotron light. If you need more resolution, we have a 5-micron resolution, space resolution. But if you use synchrotron radiation, you can go below 1 micron resolution. So you can study the growth of enamel in tooth. And that really gives you a huge amount of information in terms of life history. You can see the neonatal line of these hominids, what was the age when they died, what is the length of their childhood, and so on. And all that comes out from the growth lines that you have if you have this kind of resolution, like below 1 micron in the enamel of the teeth. And so birth, daily growth, development, and so on. Now a couple of more slides. Just we can reconstruct the brain. This is the brain of the Homo sapiens from Italy, Calabria, 23,000 years ago. This is a collaboration with the University of California even. So we are extracting the shape of the brain, and my colleagues in the US are doing the full genome of these ancient Italians. So we are trying to understand if they were different from us, and then we are going to compare them with the Neanderthals, and so on. This is a line of research that we started a couple of years ago. This is from a site called Romito in Calabria 23,000 years ago. So we are in a full ice age. And then, this is in practice my last slide, we could really try to understand when these brains that we had since 200,000 years ago, we have the same in practice brain apparently, but the mind, the new mind appeared later when, people thought that, like we saw the Chauvet pictures, the beautiful rock art that you can find in France 40,000 years ago. But the same kind of art you can find also in Australia, going back maybe 50,000 years ago. I was involved in some studies of this, but recently it was found in Indonesia 36,000 years ago. This is a Barbirusa, an animal from Southeast Asia. So everywhere Homo sapiens is present, you can find this kind of cognition kind of demonstrations, these cultures. And so we are talking about the beginning of symbolic behavior. So when we have this new light, we call it, in our brain, and really here we are really at the frontiers and very close to theology and philosophy and so on. So this is an interesting topic. So I'm nearly finished, but I just want to mention that in the last studies, it seems that already 80,000 years ago, and maybe more, there is the symbolic behavior in Africa. But so this is really the main thing I wanted to tell you. But if you want to know more, allow me to give you some publicity. This is the book that came out in Italy a couple of months ago, Homo sapiens, an unauthorized biography. And it will be published in English in a few months by Springer. And here we say things like, the new mind created imaginary realities, invented symbols, developed myths and religions, generated institutions meaning rules of behavior and so on, not just the institution, the term we use. So, then there is a new story that you can have from here. Thank you very much for your attention.
Thank you. Do we have any questions for Claudio?
Have you looked into any of the more recent evolutionary trends in humans and where those are going, or what's happening?
Well, there are recent evolution, like for example, the enzymes for digesting milk, and that was evolved. That capability was evolved about after 10,000 years ago, when we had the agricultural revolution and so on. But there is obviously a continuous evolution. But now we are replacing, in a way, biological evolution with cultural evolution. So, as you know, we have many more diseases than our Paleolithic ancestors, but we have all the medicines, or we can replace, now also even arms, or all kind of organs. So we can supplement all our weaknesses that we develop with the life that we have, since we are not anymore hunter and gatherers, with all kind of gadgets developed thanks to our symbolic thinking.
I have maybe a kind of general question. What is typically the uncertainty or the accuracy in the dating of these fossils that you find these days? I mean, using the specialized methods that you have discussed.
So, starting from a radiocarbon dating, where you can have errors, which is good only for the last 50,000 years, I said, but that is enough for studying, for example, the overlap between us and the Neanderthal. An error of tens of years. So, there is a very small error now. Optically stimulated luminescence, which you can go back to more than 500,000 years ago, you can have an error of centuries. But you are already happy if you get an error of millennia, and so on. So potassium argon also can give you errors of a few percent.
Thank you. I'll have to wind it up here. Thank you again, Claudio. And the final speaker of the session, the one standing between us and lunch, is Mario Livio. He's going to talk to us about coherent symmetric light interferometer for electromagnetic field sensing. He received his laureate degree in physics from the University of Naples, and he holds a CNR research position and was an ICT lecturer, is an ICT lecturer and consultant. His research and experimental activities are at the CNR IMM module, Microsensors and Optical Sensing System. Welcome.
Thank you, Mr. Chairman, for your kind presentation. Good afternoon, ladies and gentlemen. First of all, I have to say that it's a big pleasure for me to be here in this lecture Theater of the Faith for fascinating discussions about the light, sustained by scientific, theological, and philosophical approaches. Just 2 words about the Institute of Microelectronics and Megasystems of the CNR. The Institute has 5 local units in Italy, each of which has an advanced clean room for microfabrication and characterization facilities for optical and electrical devices. This is the summary of arguments I will discuss. My presentation briefly reviews some concepts on light from several points of view, and some industrial, scientific, and medical applications requiring accurate electromagnetic field measurements. And then other recent optical components and services for ICT, which have an optical key component in common with the electromagnetic field sensor. The advantage of optical sensing is discussed with respect to standard field sensors. And then I will discuss the optical design of the sensor based on integrated optical interferometer, giving some remarks on the terminology commonly used in the scientific literature. Well, the light has for a long time captured the attention of mankind, and several presentations of the light passed down the centuries of human history by scriptures and pictures. For instance, Egyptians and philosophers as Aristotle, Epicure, and Euclid attribute to light some kind of particles flowing toward or arising from the eyes. Now, how don't mention fascinating Asian and European novels telling about the light visions of the sea navigators watching at the bioluminescence. In some senses, a ritual rule of quarry fish species. The navigators were driven by stars and astra in the sky. Yes, it is the first global position system in history. And then in the 30th century before Christ, King Ptolemy I built one of the 120-meter tall structure called the Alexandria Lighthouse in order to confirm the importance of long-range light signals acting as navigational aids. As we will see tomorrow in the museum, also several painters from the Angelico in the Middle Age until Rubens in the 17th century attributed the entire rule to light, representing holy people surrounded by luminous hell stars or radiating golden rays. Just we recall the fascinating descriptions of the origin of life coded by the genetic code, or those described in the Bible in Genesis, light as a symbol of the revelation, as verbum, as moral behavior, enlightening humans in Giovanni, Apocalypse book, and many others. In my opinion, these are only some of the strong rules of the light for life. Of course, this is a very small part of light understanding that among many modern history scientists, Newton, Huygens, Maxwell, and so on. Perhaps we have no more time. So we just can remember that light velocity was measured more and more complex experiments just in order to confirm the relativity theories, in order to discover new physics. So we pass directly to the light applications. Here we have some great applications of light scans, just explained by their eminent authors in this conference. So about radio frequency-based applications, in the first one is the field map generated by tissue ablation. We just recall that endoluminal techniques are much less invasive techniques than standard surgical ones. So in the right image, there is an electromagnetic tomography, a recent approach aiming to implement cheap and transportable computer tomography instruments. In such images, cell aggregates, blood, hemorrhagic stroke, and tissue perfusion can be characterized. Other topical applications are based on electromagnetic wave propagation, localized services as Wi-Fi or in-fly Wi-Fi for plain tablet connectivity, or distributed services radio frequency over fiber, radio frequency over fiber, or the big data supercomputers, which CPUs are interconnected by low power all optical transceivers. And these are called the small form factor pluggable transceiver. Such optical content interconnections links can communicate terabits per second. In order the general purpose supercomputer approaches to the aggregate performance in order to exaflops. So, of course, processor performance can be improved with respect to the actual one by implementing specific algorithms by optical quantum circuits based on interferometry. So, in the picture in the left, a geographical information system of the radiating radiating electromagnetic infrastructures in the metropolitan area of Naples that I made at the early time of the mobile communication networks. Such system supports the infrastructure celebration planning and the authorization to installation in conformity to electromagnetic law limits. Now, the source density, of course, is largely increased and the electromagnetic fields are pervasive. Let's go to measurement. Far field are successfully measured by standard metal electromagnetic field sensors. So electronic circuitry is often included in the head of the field sensor to convert the field and the multiplex data to a remote unit for processing and displaying the measurement. In order to reduce the interference with fields or for use in harsh environments, some commercial sensors have fiber optic links to remote instrument, and for using several drawbacks occurs in such practical situations where these electromagnetic field sensors are generally active antennas, therefore include electronic components, PCB, power supply, and metal connection in the head of the sensor. So standard sensor may produce coupling problems in near-field measurements. For example, alterations may occur for transmitters detuning, voltage change wave ratio, or S parameter changes. These are due to the influence of the field by capacitive inductive coupling generated by the metal structure of the active sensor. So, accurate and very near field measurements is an issue for studying hotspot due to circulating radio frequency on a printed circuit board or complex electronic systems where several communication subsystems must coexist without interfering with each other. So the presence of a standard magnetic metallic sensor would produce a field influence in surrounding points when the simultaneous use of several sensors alter the field distribution in the space and in time in a given field working area, small as a printed circuit board or large as in volume of electromagnetic tomography. So the linearity and the scalability of a standard metal sensor is generally limited by a minimal frequency, wavelength to be measured, and several active antennas are required when measuring over a wide frequency range. An optical sensor overcomes these considerations because results fast and can be applied to near-field monitoring of critical terrestrial and space communication infrastructures. Also, it enables either a better power control in respect of the laws of electromagnetic field limits and the optimization of quality of service in wide area infrastructures. Well, and now the optical design. As first requirement for the optical sensor we aim is an optical design using mainly dielectric instead of metal materials in order to minimize the influence of the sensor in the Fresnel zone of the field source. For this reason, we do not require a system on chip. We do not need to integrate the sensor with CMOS technology. Therefore, we choose an external source of light supplying the optical modulator and remote light processing. We experimented lithium niobate as an optical active material for a simple symmetric refractive interferometer based on a double wide branch waveguide. The lithium niobate crystal has large second-order nonlinear coefficients and is used to implement a beam phase shift along the propagation in the arms because of Poggle effects that is the change of refractive index by application of electric field. So the interferometer is supplied with a coherent and linearly polarized light beam at 980 nanometers into the first single mode wide branch. The branching angle is very smooth, around 10 milligrad, in order to reduce the beam power loss and to preserve the propagation mode. So the beam propagation mode may be adiabatically transformed from a mode of the wave equation at the input side to the other a mode of the waveguide system at the output port. Another viable option for future implementations of integrated optics modulator include layered polymers or silicon-based metamaterials, that is the artificial assemblies of structured elements of sub-wavelength size. They have recently been used for light confinement, as shown in the right figure, and could be used for build electro-optical modulation in the next sensor implementation. Interoperability, of course, requires a choice dependent on the sensor requirements for the direction of the crystal cut and the direction of light propagation because of fixed field factors as birefringence and ferroelectricity. In the first figure, we described the integrated optic interferometer. The base component of the optical probe is a coherent symmetric interferometer developed on a lithium niobate substrate in order to exploit the Vogel effect, which results in a linear change of refractive index due to the external electric field. We can recognize in sky blue the electric substrate, an X-cut from propagating lithium niobate substrate, and sections of the optical waveguides in light green color integrated into the inner part of the substrate. The waveguides are fabricated using a higher refractive index created by carbon ions implanted into the lithium niobate. Alternative methods are used, consisting in thermal titanium diffusion into the lithium niobate or product exchange. On top figure, besides the location waveguides dark green sections are the electrodes of an electro-optical modulator, which generate the beam phase modulating electric field. The modulator has a push-pull configuration in the sense that the modulating field have opposite directions in the waveguide arms. Therefore, opposite are also the beam phase shift. We now have to remark a strange confusion that can be observed in literature, because such integrated interferometer is generally interpreted at the same way as the well-known Mach-Zehnder interferometer, but this has two detection points at 90 degrees. Each other. So where constructive and destructive interferences occur. So at the first one, a small signal modulating a large signal is detected at rest, while at the second point, a large modulating signal is detected. So the wide branch interferometer we use has only one constructive interference point. Therefore, in the detector output, we have to filter the large DC component of the signal in order to obtain a small amplitude modulated useful signal. The sensing elements acts like a transducer and supply useful signal to the optical interferometer with a minimum field performance. So we evaluated the different types of antenna electrodes, and we implemented a wideband traveling wave electrode, which provides a near ideal velocity match, differently from a classical resonant dipole, which has a smaller frequency response. Our implementation is derived by the Hooking approximation for so-called resistively loaded dipole antenna shown in the figure. And here are some questions about modeling. We proposed a device model useful for parameter optimizations and for validation of experimental data. just on the right, you can see the optical fibers toward the photodiode and coming from the laser source. Last equation estimates the output power of the modulator to the photodiode. Here is the block diagram of the setup, including laser diodes and photodetector, signal spectrum analyzer, and so on. So the source and sensor isolated or anti-inductive supports actually. The parameters of course, S11 parameters strongly depend on the wafer interface to fiber. So the optical power budget of interferometer actually provide a total loss of 28 decibel. Obtained when you accurately polish the optical interconnection. Every minimal damage in the fiber or near the grooves, pigtails, connectors require an accurate optical segment rework soldering. So in here, some tests and measures performed in a correct chamber in far field and near field are shown. Time and zero span measurements are in the upper images. We compared it with the large band commercial sensor in central photo. And on the left and right photos, there are frequency selective measurements respectively in spread spectrum and narrowband signals. In conclusion, I reviewed the purposes, the understanding, and some relevant industrial and medical applications of the light, and described an integrated interferometer optical device suitable for measurements of electromagnetic field also in the Fresnel zone, because of its prevalent dielectric structure. In conclusion, the light provides us a reference for human improvements and provides us a shining future. Thank you for your attention.
Time for questions before lunch. Anyone's got a question? It seems that lunch seems to be the priority. Thank you again. And thank you to all our speakers in this morning session. And I expect we are to reconvene at 2:15. Does that sound 2:10?
2:30.
2:10 if you are to remain on schedule for the rest of the day. So please enjoy lunch and be back on time 2:10. Thank you.
Sorry, just a small advertisement. This